We're now on section 1.2b, revisiting cost postulates from a microbial community perspective. And the first talk will be from Wendy Garrett, who will be joining us remotely.
Thanks, Karen. Can everyone hear me okay?
Yes.
Excellent. Okay, so I'm going to talk to you a little bit about Fusobacteria, colon cancer, and then wrap up with a protozoal light bite, if you will. And you should see an image in the middle of the screen there. And whether you see a mouse or a human, I hope that all of us can move seamlessly between the animal models we all use in our own labs to understand and unravel some fun, basic, and translational science. Can everyone see the mouse?
Yes.
All right, and the man? There are his eyeglasses. Next slide. So today, starting with Skip, we heard a little bit about causality theory and the importance of understanding and appreciating the role for viruses. And I think Rivers was a fantastic example of bringing that sort of science to the fore. But germ theory and thinking, I think, from the earliest configurations of the microbiome might date back to the 1400s. And so I have Fracastoro up there. Now, Koch and Heinle got a lot of credit for thinking about how the microbial... Oh, no. Okay, so in quick review, I think the early thoughts about the microbiome date back to Fracastoro in the late 1400s, and then there was a bit of a leapfrog once germ theory was formalized. And while we hear lots about Koch and Heinle, we often too commonly forget Klebs. And then once we learned about bacteria and bacterial cells, as Skip pointed out today, viruses for a long time became underappreciated. And so if we leapfrog now to Rivers and Bradford, they brought the idea of thinking about viruses to the fore when we think about disease theory. And Evans thought deeply about chronic diseases and the role of microbes in not only acute infections but chronic diseases. And I think we've highlighted that today with Jeff, thinking about the roles of microbes in malnutrition and undernutrition, which have long-term effect on development, and also from Patrice and Federico when we thought about the diseases such as diabetes and cardiovascular disease. And while we could also add Skip Version and Thad Steppenbach, although you have Skip in the room today to follow Fredericks and Relman, there was a beautiful old review that I hope you can revisit from a few years ago from Dave Fredericks and Relman about how sequencing is informing our approaches to causality theory. And the writings of all these folks, I think, are often underappreciated and worth revisiting. Next slide. Great. So I'm a GI oncologist, and I think a lot about how microbes contribute to colon cancer. I was not the first person to think about this, nor the last. But if I would bin it into three categories by which we can think about how microbes contribute, there are at least three models that are not mutually exclusive nor competitive. Single microbes can be linked to potentiating colorectal carcinogenesis. Microbial communities can act, we've heard, through metabolites today in a variety of diseases. And single microbes in concert with microbial communities in the background of host genetics, the role of the immune system, diet, and lifestyle can also contribute to the potentiation of colorectal carcinogenesis. So where does one begin? Next slide. I don't know where folks should begin, but I was very fortunate when I was starting my lab a few years ago to team up with a fantastic cancer genomicist and co-mentor a brilliant student, Alex Kostic, who is now an assistant professor here at Harvard Medical School and Joslin Diabetes Center. We found something that was initially perplexing and surprising, that Fusobacterium nucleatum, an anaerobic gram-negative microbacterium that lives within our mouth and our saliva, all of us in the audience, on our tongue and at our gingival crevice, was enriched in human tumors. We took two approaches, initially with a small cohort using deep DNA-seq and actually looking for microbial sequences in dedicated cancer genomic data sets, and then switching to a larger cohort where we used 16S ribosomal RNA gene amplicon surveys to identify an association or an enrichment between Fusobacterium and colon cancer. We found Fuso in human adenomas and human stools, but that next slide was really only an indirect association. So I think many of us are at this meeting because we approach causality using animal models, and I like mice. So I've used mouse models in a variety of different ways to approach whether Fusobacterium nucleatum potentiates colorectal carcinogenesis. And sometimes these ways have been somewhat artificial. So we took a human isolate of F nucleatum, that is Alex and Unyoung did, and they fed these APC min mice that are predisposed to develop intestinal adenomas and occasionally large intestinal adenomas, high levels, 10 to the 8 CFU per day of a human isolate of Fusobacterium nucleatum. Next slide. And what they found was Fusobacterium nucleatum, a particular type strain, in contrast with a collection of different streptococci, other oral microbes that we tested, versus just sham handling. The mice that got the Fusobacterium nucleatum developed intestinal tumors at a much faster pace than the other mice. And there's some quick snapshots at the bottom of the screen of those adenomas under the microscope and on gross. Next slide. So it looked like mice were useful. And so what we did is we took these small intestinal adenomas from mice that had been exposed to Fusobacterium nucleatum versus Strep sanguinus, and there were more in the mice that got F nucleatum, and we looked at the immune cells that were there, because as we heard from many speakers earlier on in the day, it's that interaction between host and microbe that has co-evolved over so many millennia and co-adapted, and we thought that if Fuso was working in some way to potentiate colorectal carcinogenesis, perhaps it was by hijacking the immune system. And what we found was, and the definitions and the abbreviations aren't important, the big picture here is no matter what sort of myeloid cell we could look at in these early intestinal adenomas, Fusobacterium nucleatum caused those myeloid cells, whether they were neutrophils or myeloid-derived suppressor cells or different subsets of macrophages or dendritic cells, to mushroom in population frequency and number. Next slide. And then the challenge for us has always been, just as I sort of showed you with that first symbol, if we see it in a mouse, if we can see that image in the mouse or make an observation in the mouse, how does that relate back to human tissues? And so one challenge that we've done is we did sort of the reverse. We looked at a large human data set and said, okay, we've learned something about myeloid cells. Can we see these self-same signatures in humans? And this is a human RNA-seq data set generated under the aegis of the Cancer Genome Atlas. And what we have here on the left side of the screen above the heat map is a rank ordering of these patient samples, these tumors or colorectal cancers, ranked by fusobacterial abundance. So those on the far end of the screen will have the highest level of the fusobacterial abundance. And that we've called out on the heat map here is a gene rank list of those most abundant genes that link or associate with high fusobacterial abundance. This, again, was association. It maybe supported this idea that early on there was a enhanced myeloid signature, but by no means are these cytokines and factors unique to myeloid cells. But what the mouse enabled us to do, even though it harbored a complex microbiota in these experiments, was to test specifically that Fusobacterium was a potential driver of these signatures. So we took these APC min mouse and could take particular strains of Fusobacterium nucleatum and feed those strains back to the mice and see if exposure to those F nucleatum strains increased the abundance of those genes that we observed upregulated or correlated with Fusobacterium abundance in humans. Next slide. Now, that was interesting that we saw tumors in the mouse, and it was interesting that we could repeat or replicate certain signatures that we observed in humans in mice. But at this point, circa 2014, just because there's an association between a bug and maybe a bug, or bacterium in this case, can make tumors mushroom, is there any significance insofar as human survival and prognosis? Was Fusobacterium worth doubling down? And for this, to ask this question, we moved to human cohorts using some techniques that we developed from mice, quantitative PCRs that we streamline off mouse formalin-fixed paraffin-embedded tissues. And what Kasuki Mima, a talented physician scientist in the Ogino lab, helped find out using large human cohorts is that Fusobacterium nucleatum correlated with different T-cell subsets in colorectal cancer, and perhaps even more importantly, that Fusobacterium in humans correlated with tissue and prognosis, unfortunately worse prognosis. Next slide. And then for us, one challenge that we wanted to undertake, understanding how much variety there is in different bacterial strains in terms of the metabolites they can generate, in terms of differences between Fusobacterium nucleatum strains at the gene level, was to start to generate a collection of F. nucleatum bacteria isolated from human colorectal tumors. And in the spirit of openness that I've learned from Skip Virgin and Jeff Gordon and other wonderful members like Thad Steppenbach of the microbiome community, sorry if I left anyone out, we deposited them at BEI resources. So we deposited the sequence that was generated as well as the organs so we can begin to grow the community around Fusobacterium. And the other reason to deposit these is so that as many people as possible could test and begin to phenotype these strains. And I want to talk to you briefly about how we began to phenotype these strains using mouse models. But start off, next slide. Oh, next slide. Great. In a story where mouse models wouldn't have been right. So sometimes mouse and mice are wonderful, but sometimes we would have missed the boat with mice. And so as I mentioned, we isolated a lot of CTIs, clinical tumor isolates, or a few CTIs from human colon tumors. And Hamutal, a talented MD PhD student, began to test the effects of these clinical tumor isolates on NK cell cytotoxicity. And what she found was pretty cool, that exposure of a target cell, whether it's a colon cancer cell line or an EBV lymphoma line or a melanoma line to Fuso, subsequently impairs the ability of NK cells to kill that line, raising the hypothesis that Fusobacterium, as we observed with mice and myeloid cells, could affect the immune system. Next slide. As Federico mentioned earlier today, having genetically tractable strains is really key for us to engage in mechanistic microbiome studies. And some of these strains are things like E. coli that we can all pretty much take off the shelf and genetically manipulate. Some of them are Lactococcus lactis, which is very genetically tractable. There are also some beautiful systems for Bacteroides. By and large, Fusonucleatum is not so genetically tractable. But for 23726, and because of sort of the stalwart determination of a very talented postdoc in my lab, Caitlin Brennan, and talented folks in the lab of Gilly Bachrach, we have made some inroads based on the work of Susan Kinder-Hake into helping F. nucleatum be a more amenable strain, if you will, to genetic manipulation. And because of the ability to generate transposon-based libraries, we were able to find at least 2 clones of F. nucleatum 23726 where we lost this phenotype where F. nucleatum was paralyzing the ability of NK cells to kill. Next slide. Given the biology around NK cells and given this great tool, this F. nucleatum transposon-based library originally generated in the Bachrach lab, very quickly Hamutal was able to find the Fusobacterium nucleatum protein, FAP2, and adhesin, a very large protein, 11.2 kb from this F. nucleatum, and the NK cell, or tumor-infiltrating lymphocyte gene, that FAP2 bound to. And it's called TIGIT. And for any immunologists out there, they're probably familiar with TIGIT is a really hot target these days in cancer biology and cancer immunotherapy because it's an immune checkpoint inhibitor. So what Fusobacterium nucleatum has evolved, as you will, is an adhesin, a molecule that it usually uses to stick to other bacteria that binds to and inhibits the signaling of an immune checkpoint inhibitor. And the little comment I made about mice is that actually mouse TIGIT and human TIGIT are sufficiently different at the sequence level that if we had only used mice and not cell lines, we would have missed this FAP2 TIGIT interaction. Next slide. But sometimes a lot of biology, even with the same protein, FAP2, and mice can be very useful. So Jawad is a very talented dental PhD student who was interested not in FAP2's ability to engage the immune system, but in FAP2's role as an adhesin. And he made us think about some old ideas about tumor-expressed sugars, such as gal-gal-NAC. Now, many of you are probably familiar with from the bacterial world that bacteria stick to each other. Fusobacterium is a pioneer organism in that it can help set up biofilms in the mouth, and that there are protein-sugar relationships that are absolutely essential for this bridge building or biofilm building. Interestingly, there's an old literature dating back to the 80s that suggests tumors can sometimes overexpress some of these sugars. And so Jawad took on this project where he found that FAP2, a different domain of FAP2 from what Hamutal studied, binds tumor-expressed gal-gal-NAC. And not only does this occur in human adenomas and human colon cancer, this gal-gal-NAC overexpression, but when mouse tissues become dysplastic and neoplastic in the colon, they also start to express gal-gal-NAC. And this understanding about the sugar and that it operates similarly in mouse colon tumorigenesis and human allowed us to use both mouse genetics and fusogenetics to understand that FAP2 gal-gal-NAC interaction is really important for Fusobacterium to localize and become enriched in human and mouse tumors. Next slide, please. Sometimes a postdoc gives me a wake-up call that maybe I'm becoming overly obsessed about a single organism, and that wake-up call came from Unyang, who said that maybe by being so focused on Fuso, we weren't appreciating that there might be other microbes in the tumor microenvironment that were contributing to colorectal carcinogenesis. So we asked the flip question, not only what does Fuso drive in the tumor microenvironment, but what do other microbes drive? What is similar between Fuso and other microbes in terms of the signatures that they drive in the tumor microenvironment? And that landed us on a chemokine that's far more than a chemokine called CCL2. Now, there's a huge literature about CCL2, about its roles as a chemoattractant, its potential roles in prostate cancer. What Unyang helped establish, and which mice were absolutely essential for, is that CCL2 is a skewing factor for myeloid-derived suppressor cells, and it leads to enrichment of myeloid-derived suppressor cells in mouse colon tumors. And having mice where we could pull apart that tumor microenvironment and then actually inject cell types in, do add-back experiments with myeloid-derived suppressor cells and tumor-associated macrophages, and begin to understand the relative contributions of different myeloid cells to tumor growth and spread was totally enabled by the mouse, something we could never have done in cell lines alone or in human tissues. And so CCL2 and that story in terms of the role of myeloid cells we're deeply indebted to for mice to understand the critical role of MDSCs in particular. CCL2 is a skewing factor, and then the effects that MDSCs med out on T cells. Next slide. So I think we all have our favorite organism. Maybe it's a genus, maybe it's a species, maybe it's higher up on the taxonomic tree, maybe it's not a bacteria, maybe it's virus or phage. But I never want our consideration of bugs to be sort of polar, and I never want certain organisms to be binned into that sort of singular pathogen category. And so when I conceptualize about Fusobacterium, which is a symbiont in all our mouths and by and large doesn't cause trouble in our mouths, I want us to sort of think about Fusobacteria and cancer from a live and let live hypothesis. And what I mean is that Fuso is just searching for a warm place, if you will, with adequate carbon sources to live. Unfortunately, it ends up in the wrong place at the wrong time for some mice and more importantly for some humans. It can recruit and recondition immune cell types via CCL2, but that's not a unique feature to Fuso. The other thing Fuso can do is that it can inhibit immune cell-mediated killing of bacteria. Many bacteria have different strategies to evade the immune system. That's not unique to Fuso. But that Fuso has an adhesin that engages an immune checkpoint inhibitor is kind of curious and points us to potential targets for colon cancer and also for thinking about microbial actors in disease states and health. The other thing that Fuso has is not only one bacteria protein, but a bacterial protein or adhesion with multiple functions. So FAP2 seems perhaps particularly unlucky for some folks insofar as it enhances tumor adherence and enrichment by a sugar relationship. And this provides us with some different thoughts and solutions potentially for preventative strategies for people with adenomas and cancers of colon cancer. Next slide. So sometimes it's okay to think about a single organism, and I'd say sometimes it's okay to seek, and sometimes it's okay not to seek. And for people not with the visual, not with the audio and not the visual, I'm abbreviating seek, not S-E-E-K, but seek for sequence. So there's a time to use mice, there's a time not to use mice, perhaps to use organoids. There's also a time to seek with some projects and not to seek with some projects. Next slide. So what do I mean? So I'm going to start off with a summary slide in the interest of time, but real briefly, an intrepid postdoc who's on the job market by the name of Michael Howitt got interested in Tuft cells, taste-chemosensory cells that exist throughout our body but are curiously in our small intestine and colon. And he was curious about what they taste. And what we found something that they tasted, and we're quite surprised. It was a symbiotic parasite that was terribly underappreciated and understudied, but widely present in the microbiota of conventional mice. And what Michael helped unravel was a pathway by which the epithelium senses a broad variety of parasites, protozoal and helminth, and engages type 2 immunity. Next slide. So how did we find this, and why wasn't it time to seek? So there are many kinds of epithelial cells, as many of you know, in the intestine, absorptive enterocyte, goblet cells, enteroendocrine cells, and Paneth cells. Tuft cells are these peculiar cells with this wonderful apical tuft, and these neat vesicles have been underappreciated roughly till 2015, 2016. Next slide. And so we began to study them using markers like GFI1b, discovered by Philippe Jay, and also by DCLK1. And we found that the mice here, bred in-house, if you will, had more Tuft cells than had ever been observed in the half a dozen papers published on them. But when we brought in mice from Jackson or other vendors, there were many fewer Tuft cells. This is a similar theme observed by many laboratories, and it also often invokes the knee-jerk reaction of, it's the microbiota. Next slide. And so we performed an experiment that many people in the audience have undoubtedly performed, and that's the feeding the fecal contents experiment to demonstrate that there was some role in causality of this observation for the microbiota. And we found that, indeed, there was something in the fecal contents. Next slide. And what that it factor was this little guy that I've already introduced you to, Trichomonas mirus. So before seeking, even though we were all ready to do a 16S ribosomal RNA study, what we saw using our eyes, the microscope, in the sort of spirit of Ramoni Cajal, was that we had a lumen full of Trichomonas mirus. And as you know, these are eukaryota parasites, so if we had looked with 16S, we wouldn't have found them. Next slide. We were able to isolate these in pure culture, even though we had to work on developing a media, and they were sufficient to rescue the number of Tuft cells or cause them to mushroom to superphysiological levels. Next slide. And it wasn't just a symbiotic parasite, but Tuft cells were responsive to a host of, very genetically divergent helminths or other kinds of parasites. Next slide. And so now what we're doing, having defined sort of this downstream limb of the pathway that Tuft cells are huge producers of IL-25, we're going upstream. And since Tuft cells are taste-chemosensory cells, we're very busy trying to find the taste receptors that Tuft cells use to taste protozoa and helminths, and they appear to be different taste receptors, but the downstream signaling seems to be shared. So for us, mice are sort of this fantastic system by which we're continuing to learn and unravel different elements of the host microbiota interaction. I think mice continue to offer us endless opportunities, sheer wonder, and delight in science. So now for welfare. Next slide. Okay, for me, my lab, and many of the people in the audience, mice are a sine qua non, without which not of our research. And I think often animal protocols and monitoring are constructed around looking for signs of distress secondary to procedures. But often our mice, whether the APC-min mice or other genetically mutant mice like the MSH2 mice, develop disease, and we need simple workflows for monitoring them. And for us, sometimes we use something as simple as a sticker to mark them. So simple workflows to minimize the labor but maintain the health of the mice. I think we need simple methods for recognizing signs of suffering and taking action. And sometimes it's as simple as just changing the bedding. Many facilities use corncob bedding, but often there's paper-based bedding that is much more gentle on an animal that's developing prolapse from tumor. And I think we have to refine humane monitoring and endpoints. Sometimes we've used invasive monitoring, but that's tough on mice with colonoscopy. And more recently, we're trying to set up systems to non-invasively monitor our mice, and these are often luminescence-based in vivo imaging systems. Last slide, and it's the challenges. So innovation, future directions, and challenges. I don't know about humanizing a mice, but I like the idea of building a better avatar. I didn't come up with this idea. It's a term very frequently used in terms of mouse models in the cancer biology world. But our making a better avatar can come from defining the microbiota, as we heard from Federico Rey, in terms of defined mouse communities, or using communities from humans, as Jeff Gordon spoke about. In terms of cancer biology, we can better define tumor genetics and hasten timelines. This integrates the organoid system that Vince mentioned. So we can genetically perturb organoids with CRISPR-Cas, and we can couple these with orthotopic colonoscopy-guided transplantation. And if you're curious about this, Omer Yilmaz has published a paper in 2016 outlining some of this. In this way, I think we can do more effective tumor and tumor immune monitoring and also assess perturbations, be they diet, which we've heard about today, drugs, or immunotherapy. And I'd also like to endorse a concept that Skip started off the meeting with about openness and sharing of models and methods. It's so important for improving our reproducibility across facilities, within a university and beyond, and also advancing the pace of scientific discovery, and ultimately, in terms of cancer, helping patients. So with that, I apologize for the technical snafu in the beginning. Last slide. Next slide. And I just want to acknowledge the past and present members of my lab, all our wonderful collaborators that enable our research in ways that I could never have imagined, and our funding, current and past. Thanks so much.